The next 1 hour and a half to her excellent speakers talking us about autophagy, stress, and cell death. We will start with Patricia Boya. Patricia is now in the SEBC in Madrid. She is a group leader, and her research line is fully dedicated to autophagy. So she is going to talk us about autophagy, metabolism, and cell fate.
Good morning, everybody. Thanks, Isabel, Miguel, and Alberto for the invitation. Hear me now? Hello? Okay. So I would like to thank the organizers for inviting me to be here in this, no? Hello? Can you hear me? Oh. Hello? Okay, nice. So I thank the organizers for inviting me to this amazing conference. And as Isabel told you, my lab is interested in autophagy, what are its roles in cells, and how alterations in this pathway has pathological consequences. So as you know, there are two main catabolic pathways in cells, the ubiquitin proteasome system and the autophagy system, where degradation takes place inside lysosomes. And autophagy can be divided in several pathways depending how material gets to lysosomes. The most known pathway, the one I'm going to be talking about today, is macroautophagy, and this process starts with the formation of a double membrane that closes and engulfs inside parts of the cytoplasm, including whole organelles, and this autophagosome will fuse with the lysosome. During microautophagy, contents are directly translocated inside the lysosomal lumen, and sharp-permeated autophagy is another selective pathway of autophagy where proteins that harbor specific motif are targeted to lysosome. Inside lysosomes, the material will be degraded, and most importantly, this can be recycled and can get back to the cytoplasm to sustain cellular functions. We know now that autophagy can be highly selective, and I will go more deeply into this story, but we can degrade peroxisomes, ribosomes, and of course mitochondria. And there are several pathways to degrade mitochondria. It's a pathway used to degrade damaged mitochondria, but it also can be a pathway to eliminate mitochondria when they are not needed. And the most known pathway for mitophagy is the pink parking pathway that modify mitochondrial outer-membrane proteins, and this allows the binding of P62 to LC3 and form the autophagosome around the damaged mitochondria. But there are other pathways going through mitophagy receptors, for example, BNIP3 and NIX, and I will be talking a lot about NIX, where these bind directly to LC3 and help the formation of the autophagosome membrane around the mitochondria. So all cells have some degree of basal autophagy, but this process can also be induced. And as I told you, it can degrade many things. It can degrade organelles and protein aggregates. It is very important also to degrade intracellular pathogens and viruses. It's also an essential response to protect DNA damage and also has roles in cell death. And it's also an essential metabolic adaptation to sustain cell viability during starvation. So looking at all of these important functions in cells, it's logical to think that alterations in autophagy has many consequences for many diseases, from neurodegeneration to aging to cancer and to diabetes. But today, what I'm going to talk to you about is how we think autophagy is important for development and differentiation. So we know that it's important to development of erythrocytes, adipocytes, macrophages, and B cells. And we think that it's important to remove organelles. It's also essential for cytoplasmic removal, but it also plays a role in cell death. But when we started this pathway, this project, it was not really known what autophagy is important for neuronal differentiation. And we tackled this process by using mainly autophagy deficient mouse models. So in some preliminary studies that we, using olfactory bulb neural stem cells, when we isolated these cells from the mouse, the GFPLC3 mouse, that is a mouse that allows us to see the formation of autophagosome. When we put these cells differentiated into neurons, we observed that the cells that differentiated in culture, here stained by B32 tubulin, were the ones that had autophagosomes. And when we isolated these cells from autophagy deficient animals, for example, the ATG5s, we observed that in vitro differentiation reduced the number of differentiated neurons, suggesting the idea that autophagy is essential for neuronal differentiation. But we wanted to tackle this process in a more physiological context, which is the retina development. So the retina is the light-sensitive tissue that lays the back of our eyes. It's composed with many different cell types, for example, rods and cones, which are the photosensitive cells, and makes synapses with other cells, and finally to retinal ganglion cells. Retinal ganglion cells are the only projecting neurons of the retina, and their axons from the optic nerve. So retinal ganglion cells are the first cells to differentiate in the mouse retina, and our retina as well. And we wanted to see whether autophagy was important for differentiation of these cells. And this is just a section in an embryonic retina at 50.5, which is the peak of a differentiator of retinal ganglion cells. So this is, we work mainly with mouse, and we isolated the retinas from different embryonic embryos at different stages. So this is the retina, and the retina has several advantages, because we can culture the retina in explants, and this is particularly important for autophagy studies because we can manipulate the culture conditions and the nutrients in culture, and we can see the autophagy response of the retina. It's also very nice to culture the retina in explants because you maintain the 3D structure of the retina, you maintain cell-to-cell communication as well to cell-to-matrix communication. And this is very important. And we know from studies in the lab that putting the retina in culture, the embryonic retina, the developmental process carries out as in vivo. Of course, we can do confocal microscopy, flow cytometry, blood, so it's a very useful model to study neurogenesis. And this is to give you just an example. So these are retinas isolated at different embryonic stages, 13, 15, and 18, and they are stained with the brain 3A. This is a transcription factor specific for retinal ganglion cells. And we can see that the development of the retina of retinal ganglion cells starts around 13.5, and this is just the wave of differentiated cells. They are extended in the whole retina at 15.5, and neurogenesis in retinal ganglion cells is completely at 18.5. So we wanted to study the role of autophagy in these retinas and use wild-type and ATG5 deficient animals. And we observed that during this period of neuronal differentiation, we observed that wild-type retinas, after 15 and 18, there was an important reduction in the number of mitochondria. This is mitochondria stained with mitochondrial automembrane protein TOM20. However, this mitochondria decrease was not observed in the ATG5 deficient retinas. This is just some flow cytometry plots, and this is the quantification. You can see that wild-type reduced mitochondrial mass, while mitochondria retain in the ATG5 deficient retinas. More importantly, these deficient retinas had alterations in the number of retinal ganglion cells, here stained by brain 3A, so reduced number of brain 3A neurons. The phenotype was also very clear when we stained the retinas with B32 tubulin, and as you can see, there were important alterations in the axons, in the morphology of the axons, in the ATG5 deficient retinas. We could mimic this deficiency in neurogenesis by explants, using retinal explants by using classical autophagy inhibitors like 3MA and cyclosporin E to block mitophagy. You could see that culturing the retina with those components increased mitochondrial mass and also reduced neuronal differentiation. More importantly, when we induce the process, when we induce autophagy with rapamycin, we could see an important decrease in mitochondrial mass. This is flow cytometry, also by immunostaining, you can see an important decrease in the number of mitochondria. This process was blocked by blocking 3MA, but this was associated with an increase in the number of differentiated retinal ganglion cells, as you can see here, and this was blocked by 3MA. So this data suggests that proliferating neuroblasts have a lot of mitochondria. This is reduced in the young retinal ganglion cells when differentiate, and this process is regulated by mitophagy because rapamycin is increasing and ATG5 is decreasing. But in order to understand more the molecular mechanism of this mitophagy, we checked the expression of these mitophagy regulators. And we did the quantitative mRNA expressions, and we see the pink and parking, two classical mitophagy regulators. Their expression was regulated, but not at the embryonic stages, but later in the adult. However, NIC's expression was importantly upregulated during the period of neuronal differentiation, and this could be an indication that maybe NICs was in charge of this mitophagy during neuronal differentiation. So we took advantage of the nix deficient animals. This is an animal that has, so they are viable, they are born, and their most important alteration is that nix deficient erythrocytes display mitochondria in their cytoplasm. As you know, erythrocytes are the only cell in our bodies, or the few cells of the inner body that doesn't have mitochondria. So nix deficient erythrocytes have mitochondria retained. And this is through a process of programmed mitophagy that occurs normally in erythrocytes. So we look at the phenotype of the retina of these animals, and we could see that in a similar way of ATG5 deficient animals, nix deficient retinas have increased mitochondrial mass. These are retinal flat mounts. This is retinal sections. You can see an increase in the level of a number of mitochondria. And again, we had a defect in the neurogenesis. So this is retinal sections stained by B30 tubulin, a massive decrease in the number of retinal ganglion cells, also stained here with brain 3A and gamma-synuclein, two specific markers for retinal ganglion cells. You can see there was a massive decrease in the number of retinal ganglion cells. This phenotype was retained in the adult mice, and you can see that the nix adult retina also displayed increased mitochondria and decreased neuronal differentiation. So we could play nix in our scheme, and we can say that this mitophagy that occurs during neurogenesis is ATG5, of course, and nix dependent. So this was kind of strange because we know that during cell differentiation, what normal happens in many cell types, or most of cell types, is that differentiation is associated to increased mitochondrial biogenesis and increased mitochondrial activity. And in our case was the contrary. We also know that in a reverse process, so during reprogramming to pluripotency in pluripotent stem cells, this reprogramming process is also associated to a decrease in mitochondria, a decrease in oxidative phosphorylation, and increase in glycolysis. Other paradigms of metabolic reprogramming occur also, for example, during oncogenic transformation, and we know that normal cells rely mostly on OXPHOS. However, cancer cells rely more on glycolysis, and this is the known Barbour effect. This metabolic reprogramming also occurs in macrophages. When macrophages are polarized to the M1 pro-inflammatory phenotype, they rely mostly on glycolysis, while M2 macrophages, the M2 more anti-inflammatory, rely on OXPHOS. We also know that this metabolic reprogramming occurs during hypoxia, and during low oxygen, cells degrade by mitochondria by mitophagy. This is known, and they also change the metabolism towards a more glycolytic phenotype. So we have in the retina that there is a decrease in mitochondria, and we wondered whether this neurogenesis was, so we wanted to understand if this neurogenesis was also associated to a metabolic reprogramming towards glycolysis. We also wanted to understand if mitophagy was regulating this metabolic change, and we also wanted to see whether manipulating metabolism, we were able to manipulate the differentiation of retinal ganglion cells. So we first evaluated the metabolic profile during neurogenesis, and we could observe these are seahorse experiments in embryonic retinas. Can you imagine we have to put many retinas in the well in order to do these experiments? We could see that the lactate production or extracellular acidification rate was increased in the stages where we could see neuronal differentiation, and it was reduced afterwards. We also did some metabolomics assays, and we also could see that during the process of neuronal differentiation, there was a peak in the levels of many glycolytic regulators, like lactic metabolites, like lactic acid and pyruvate. We also did transcriptomic by doing RNA sequencing, and we could see that, again, the metabolic change towards glycolysis was confirmed with a peak in the expression of many glycolytic regulators. So we would see that it was a glycolytic switch during neuronal differentiation that was reduced afterwards. So it's a peak that occurs at the same time that retinal ganglion cells are being differentiated. The next question is that, is this glycolytic switch regulated by mitochondria, by mitophagy? And the answer is yes. So we look at the MR expression of in the ATG5 deficient retinas, and you can see that these retinas have reduced expression of many glycolytic enzymes. They also have a reduced in the lactate production. This phenotype was also observed by using retinal explants, and we could see that using 3-MA and cyclosporine A also reduced expression of all these glycolytic enzymes and also lactate production. And this phenotype was also observed in the nix deficient animals. So this data indicated that mitophagy was upstream of the glycolytic switch, and it could be regulating metabolic reprogramming towards glycolysis. Can we modulate differentiation by modulating metabolism? And the answer is yes. So we did in the retinal explants, we blocked glycolysis with 2 different drugs, and we could see an important reduction in the levels of differentiated neurons. And doing the opposite experiment, blocking the pyruvate entry into the mitochondria that increases glycolysis, resulted in increased neuronal differentiation. More importantly, this increase in neuronal differentiation was independent of autophagy because blocking with autophagy did not decrease the number of retinal ganglion cells, suggesting that metabolism is downstream of autophagy. So we can now put a pathway together, and we have shown that neuroblasts have a lot of mitochondria, young retinal ganglion cells have reduced mitochondria, and this is due to a mitophagy effect dependent on NICs that is regulating a metabolic change towards glycolysis, and we know this is the case because we can modulate the process and see what is upstream and what is downstream. As I said, there is another paradigm of glycolytic switch. This is macrophage polarization. So in order to see if this mitophagy regulation of metabolism was general and it could happen in other contexts besides the retina, we used the paradigm of differentiating macrophages towards the pro-inflammatory phenotype by incubating them with lipopolysaccharide and interferon gamma, and we know these macrophages rely more on glycolysis, or by incubating them with interleukin-4 and interleukin-13 towards more an anti-inflammatory process. And we evaluated autophagy and mitophagy during this process. The first thing that we observe is that M2 macrophages have reduced mitochondrial mass in comparison with M2 macrophages. And when we manipulated autophagy in vitro by using cyclosporin A or 3-MA, we could see that the mitochondrial mass was increased in M1 macrophages, but this didn't have any effect in M2 macrophages, suggesting that mitophagy could be active in M1 macrophages, but not in M2 macrophages. More importantly, this manipulation of autophagy reduced the expression of glycolytic enzymes in culture. So you can see that 3-MA and cyclosporin A block the expression of many glycolytic enzymes, and this was also for using macrophages isolated from the NICs deficient animals. So NICs deficient, this is the expression of NICs in the M1 macrophages. So these macrophages that carry out mitophagy have a lot of NICs. M2 macrophages have reduced levels of NICs, and the macrophages that do not have NICs have reduced expression of the glycolytic enzymes, suggesting again that M1 macrophages need mitophagy to have this glycolytic profile. More importantly, this also has consequences at the inflammation level, and you can see here that incubating these macrophages with 3-MA and cyclosporin A reduced the expression, the MNR expression of many pro-inflammatory cytokines, and also this was observed in the NICs deficient animals. So this confirmed our hypothesis that mitophagy or NICs-dependent mitophagy is essential to do metabolic change, and in this case was regulating the generation or the polarization of to M1 macrophages. So going back to our retina, this is our working model. So we think that during neurogenesis, there is a mitophagy, that's a process that eliminates mitochondria. The cells are forced to do glycolysis, and this is essential for neuronal differentiation. But this is a bit strange because we know that retinal ganglion cells, adult retinal ganglion cells, need a lot of mitochondria. So we postulate that there is a lot of biogenesis in the later developmental process. Why would a retina do such a complicated pathway? So we have several hypotheses. The first one is that this could be a mitochondrial quality control. So during these cells proliferate a lot. They could maybe have damaged mitochondria. So the cell, by eliminating the damaged mitochondria, and then proliferate and then make the ones that are healthy perform biogenesis, maybe this could be an idea that this neuron will start its life as a differentiating neuron with only healthy mitochondria. So we are currently exploring this hypothesis. There is another hypothesis we are also trying to understand. Maybe this metabolic change towards glycolysis is to meet the anabolic requirements for cell differentiation. And we know that this is the case, for example, during macrophage polarization and during oncogenic transformation. Those cells use glycolysis to have anabolic intermediates to keep proliferation in the case of cancer cells and to sustain the immune response in the cancers of macrophages. So we are also exploring these two hypotheses. So with this is just my last slide. So I hope I convince you that NICs-dependent mitophagy degrades mitochondria during neurogenesis, that this results in a metabolic switch toward glycolysis that is essential for cell differentiation, and that in macrophages, mitophagy also regulates glycolysis and the pro-inflammatory cytokine production. And with this, I would like to finish. I'd like to thank especially Lorena Esteban Martinez, a very talented student in my lab that now is doing a postdoc in CENIC. She's here in the audience. She's done most of the work I've shown you today. This is my current group, of course, of collaborators and or finding funding sources, and of course, yours for your attention. Thank you very much.
Yes, a beautiful work, really congratulations. Sorry for my ignorance. It is in the differentiation happening in the perinatal period, or because this drop in mitochondrial mass resemble beautiful work done by June, April in liver, that for mammals, that they are going to have an anoxic shock during delivery. They really reduce very much the mitochondrial mass in the liver, and they resume the biogenesis very quickly after they really, in the first hour after that and over the next week. The suggestion is if this resembles that, it might suggest a way to protect the cells for a really very ischemia problem that we have in delivery, and might be one of the third hypothesis to test.
Okay, yeah, this is a very nice, interesting observation. So the data I show you today is everything is embryonic, so it's from 12, 13, 15, 18, so it's embryonic. And I didn't show you the data because I didn't have time, but we think that the signal that triggers this mitophagy is hypoxia. So we have some data suggesting that there is hypoxia during retinal differentiation, and this triggers NIX expression and another pathway, mitophagy and the metabolic profile. So in this way, could maybe resemble one of the things you are suggesting. But what we think as well is that this is a way of maybe eliminating the type of mitochondria. For example, in the heart, it's been shown that there is a similar process of mitophagy. In this case, it's a pink park independent process, and this is during cardiomyocyte maturation, and they change the mitochondria. So the perinatal mitochondria are very elongated, and then they mainly catabolize, they mainly use sugars, and the adult mitochondria in the heart mainly use fatty acids. So in their system, it's a way of changing the type of mitochondria.
Actually, I was wondering whether you think that the byproducts of the glycolysis might play a signaling role in differentiation. So, for example, if you would take the ATG minus cells that don't get rid of the mitochondria and then add back the byproducts of glycolysis like lactate and things like that, and ask whether this has a positive effect on differentiation.
Yeah, we have not done experiments, but it would be easy to do and it would be very interesting. So thank you for the suggestion.
No more questions? So, Patricia, my last question. I understand that you consider mitophagy as a potential mechanism for cancer cells to switch metabolism, or is something more related to development or other physiological processes?
Yeah, this is a very interesting question. We have not addressed that in cancer cells. But again, this would be easy experiments to perform. So, and the role of autophagy in cancer is complex. So it's been suggested that it could have proto-mutagenic, depending on the stage of the tumor, it could be pro-tumorigenic or anti-tumorigenic. So, but in vitro, this would be easy to test. So it would be not very complicated to see if mitophagy is also, so eliminating mitophagy in cancer cells will reduce their glycolytic expression. We have not done those experiments. Yeah.
No? So thanks very much, Patricia. And we move on to the second speaker. Our second speaker is José Antonio Enrríquez. He is actually in the CENIC, in the Center of Cardiovascular Research, National Center of Cardiovascular Research in Madrid. He is their food professor and group leader. But in fact, he started his research line in the University of Zaragoza, where he got a full professor position. And after that, he moved to Madrid. His research line is mainly focused on mitochondria, everything related to mitochondria, but particularly how genetic variability affects mitochondrial function and the structure, research about the structure of the components of the respiratory chain. So, Toño, thanks very much for coming. Thanks very much for accepting our invitation.
Okay. Thank you, Isabel, and thank you, Alberto, and Miguel, and all the three societies. It is really an honor to be in these meetings. I not belong to any of the societies. I might think my mind afterwards. Because I never finish on time, and well, I finish on time, but I never end to the last slide. I really decided to present the people who really deserve the merit in advance. I'm going to present results that are coming from the last, I will say, 10 years of work in our lab. So it was started with Raquel Moreno Los Huertos, that is now assistant professor in Zaragoza University, followed by the thesis of Ana La Torre, the thesis of Ana Victoria, and a fundamental contribution in the proteomic by the group of Jesus Vázquez in the person of Enrique Calvo. There are many collaborators and many important collaborations that participate along these years, and probably I forget some. If I do, I'm really sorry. I didn't mean that. So I'm going to give you only two take-home messages that I think are very important because I noticed that some students and many PIs get a little bit fed up about mitochondria because mitochondria is now appearing everywhere. It was not the case when I started almost 25 years ago. We were just a few bunch of people working in something that was very relevant, and was very relevant because in the time of genomics, those are scale size of the chromosomes of a human cell, and this is the mitochondria. And actually, this doesn't make the merit that should be. This is even smaller, but I don't have size letters of the same of the scales to put it. So you can calculate in size mitochondrial DNA and notice that this is a logarithmic scale, represent a ridiculous amount of DNA in terms of size. So this is because yes, only a bunch of people kept working on this for many years, because it should be irrelevant due to the size. However, cells do not have the same thinking, because if you just change the perspective, you see that in terms of mass, I'm talking about not base pair, but mass, mitochondrial DNA, the chromosome of mitochondrial DNA represent as much as many other chromosomes. That is the first thing that is not everybody aware of it, but I think that is very important to remember. But if you go to a transmission oocytes, mitochondria is the more represented in terms of mass of any other piece of the genome. Now the question is why? Why the cell, despite our belief, think that mitochondrial DNAs deserve such much effort in terms of mass and therefore in terms of energy? So this is one thing. This is the first take-home messages. So the cells consider mitochondrial DNA much more relevant than scientists. So then the mitochondrial DNA is odd and is really not a very good, easy to handle piece of DNA because it is extranuclear, it's polyploid, has a clonal inheritance, and has a higher mutational rate It doesn't suffer recombination. So how, despite of these complications that all the replication machinery has, still we keep such amount of effort in mitochondrial DNA? So I think that this is coming the second take-home message that I want over the next few slides, just why we figure out that this could be the case. So mitochondrial DNA encoded only 13 proteins, although it has 37 genes, but the rest of the genes are dedicated to make these 13 proteins. And all these proteins participate in a single process that we call oxidative phosphorylation, or OXPHOS. But they are not making it. They are only making subunits that participate in it. In particular, 7 subunits of a big nanomachine that is called complex 1, that is made out of 44 different subunits. 1 subunit of the complex 3 that has the majority of the subunits also encoded in nuclear genes, 3 subunits of complex 4, that, and then 2 subunits of complex 5. So still, these genes do not make a single of the nanomachines. They only participate of subunits, and it creates a difficulty that I'm gonna think that is why we keep the mitochondrial DNA. First, the first difficulty is that these 13 proteins suffer uniparental inheritance, do not generate variability by recombination. They do it by a higher mutational rate. And important that we also don't consider, they are identical for all cell types because we have a clonal expansion of few copies. We don't have the possibility to have variants in different tissues. On the contrary, the 76 proteins of nuclear DNA of these complexes, they generate variability by recombination and biparental inheritance. They have lower mutational rate, and we have tissue-specific variants. So when you make a newborn, or when you make a new embryo, you have really to be sure that these proteins match with these proteins in the beginning, when the cells differentiate, and when the cells really express variants. And also they should do that with the genes coming from the mother, that they already that they work, and with the genes coming from the father, that they don't know if they are going to match. Because the match has to be physical, not only functional, because they are building blocks of engines or nano-engines that are called respiratory complexes. So, a second thing that is, we always believe that mitochondria, and we teach, and it's my fault as well, which is the mitochondria is the powerhouse of the cells. And this is a very interesting and powerful concept, but is also hiding probably more relevant function of the mitochondrial respiratory chain than just making ATP. So there are 3 processes in this structure, and this is true for our mitochondria in mammals, but this is not true for mitochondria in other eukaryotes because they have additional proteins that can really give electrons to CoQ and take electrons to CoQ without pumping protons. This is lost, this capacity is lost in mammalian cells. So electron transport is and ATP synthesis and proton pumping are the 3 elements that are common, and it's the processes that are really tied to this structure. So electron transport is required for, of course, take electrons from different processes in the cell, metabolic process like beta oxidation, glycolysis, a carbon metabolism that is very important, but also uridine synthesis or pyrimidine synthesis, and cells that doesn't allow to recycling CoQ kind of make the building blocks for transcription or the building blocks for replication. And there is in this process also there are derived elements like rocks, but considering them here as a signaling molecules. Proton pumping is a consequence of the electron flux in particular complexes 1, 3, and 4, and this proton pumping is used for many transport activities that depend on the membrane potential in the mitochondrial inner membrane, and ultimately also for ATP synthesis. However, you can imagine that some cells that require, for example, very anabolic capacities, and they derive the ATP synthesis to ATP by glycolysis, they still need the recycling of CoQ to make pyrimidines and to one-carbon metabolism. So they are more prone to electron transport, but they are not as interested in proton pumping or ATP synthesis. So, but we have to use the same machine in cells that require more way of electron transport, less way of ATP synthesis, or the other way around. We have the same system. In addition, all these systems participate in regulating several parameters that we know that are critical for regulation, for example, the activity of the acetylases, and therefore the activity of epigenetic, the activity as well of HIF-1 alpha pathway, for example, and then the adaptation of the metabolism. And this is actually modulated by the work of this system. So, because a liver cell uses mitochondrial oxygen for one purpose different than muscle cell. And if you go to brown fat, they don't care about ATP, they only make heat. So we got to the conclusion that genetically we cannot define and optimize oxford system. So there is no way that by selection we have an oxford system good for all and every pathway that we want to use. This is the second take-home message, and this is the two take-home messages that I want to put here. So now the rest is going to be just trying to support these ideas, okay? Then how we get the oxfords that we need? There is only one way. We make an oxford that is not damaged, that will be even, and then we fix it by very regulatory mechanisms, the oxfords that we need for a particular metabolic situation, for a particular cell type. So, and then housekeeping no longer means everything is equal in every cell. It's actually multi-layer regulation secret that need to adapt the housekeeping. And I think that this message is very important for this audience. So despite being expressed everywhere, they have to be tightly regulated to make the cell healthy. So, and this is the overall idea. We have to have circuits to really read out which oxfords we have in each cell, and if this oxford is the one that we need, and then we have to have sophisticated mechanisms to really, after reading that, adapting expression of nuclear genes that modulate the oxfords to make the oxfords that we really need. So, second idea that you already know is that there is an enormous variability in mitochondrial DNA in human beings. Each spike represents one sequence of one mitochondrial individual, and they do, because they do not recombine, they can really be organized not only in haplotypes but also in haplogroups, and then end up to know that at least mitochondrial DNA in human beings is monophyletic. It can go up to one woman that lived in Ethiopia 140,000 years ago. So this was started by populational genetic analysis, and the intrinsic assumption was that all these variants were phenotypically silent. There are several groups, among others, just questioned this idea, finding association with several diseases or several phenotypes in the different variants of mitochondrial DNA. But molecularly, we couldn't demonstrate functional differences between the different two haplotypes. So there was a collision between populational genetics and molecular biology or cell biology in the interpretation of the results. So we thought that if our idea, our two ideas that we put further before are true, then we can really have wild-type modulation of the function of mitochondrial DNA having different variants of mitochondrial DNA. So then we generate cells with the same nucleus, but different mitochondrial DNA, in this case for mice, to see if they were different. And the answer in terms of oxidative phosphorylation was they are not different. So they are respiring equally, and this is a control in which we have a mutation that makes the complex one non-functional. Raquel did this analysis for many, but she was very persistent, and she was just checking for different types of phenotypic consequences. She found the following. Some haplotypes has produced basically more ROS than other haplotypes, and this was really correlated with having more mitochondrial DNA per cell. So see, this was a minor difference. But she really wondered, because all of them are wild type, is this really meaningful in terms of function? And she really was able to equalize the ROS production with a NAC of tyrone. And when she did that, she equalized the mitochondrial DNA content. So if you eliminate the ROS signal, then the cell do not distinguish the haplotypes and have the same amount of mitochondria. And what was the consequence? Now, a different respiratory capacity was revealed. So we failed for many years to distinguish differences in oxphos function because we didn't thought in the mechanisms that recognize the difference, react, and then put the mitochondrial oxphos to the level that the cells require, even activity of the oxphos. So this was done in cells and might be really not be related with what happened in bodies. So the next step that took several years to go is to prove it, to do the same experiment, nucleus identical, different mitochondrial DNA, but in that case in the animal. So this is what is called complastic animals. By process took 3 years to generate because we went over 20 generations to be sure. And we used 2 haplotypes and the nuclear background of black 6. So by next generation sequencing, we really could demonstrate that it's actually the same nucleus, but different mitochondrial DNA when we started with a completely different nucleus. So these guys are different, as littermates are different with the same nucleus, but they have some one DNA and some other DNA. So we have the tools to evaluate if what Raquel found was correct. So what is different between the 2 DNAs? These represent the respirosome that I don't have time to explain, but this is respiratory complex 1, the dimer of complex 3, and complex 4. And in here, this dot represent the differences in sequence of the proteins of this. I'm going to take away all the nuclear encoded proteins that are not related to the mitochondrial DNA, and now you have the respirosome with only the proteins encoded in the mitochondrial DNA, and you see that all these dots represent the ways that were difference between the 2 mitochondrial DNAs. They are all in the surface of the proteins in areas that interact with other proteins encoded by the nuclear DNA. This is just a nice idea. This is complex 1, this is the dimer of complex 3, and this is complex 4. Okay, the first thing that now Anna tried to do it is just to check if the only difference that we could detect in the cells has been able to detect in the animals. So for that, during the period to generate the complastic, appeared a tool developed by Mike Murphy in Cambridge, in which he was able to measure in situ, in vivo, hydrogen peroxide in mitochondrial matrix. It was a very nice trick. He has a molecule that crossed the membrane, you inject it to the animal, cross the plasma membrane, and cross the mitochondrial membrane, accumulate 2 orders of magnitude more in the matrix than outside. This molecule is made in a way that in the presence of hydrogen peroxide, it breaks. So you inject the molecule, you wait 6 hours, you kill the animal, extract the tissue of interest, and you calculate how much of the molecule that you injected was broken as an index of how much hydrogen peroxide was the basal conditions in vivo in the cell. So you didn't manipulate anything before the analysis was done. So this is what is represented here. So in liver or in heart, the proportion of the molecule that was broken between same nucleus, the different mitochondrial DNA, and you see from here to here in both tissues, you have more broken, that means more ROS. It was too good to be true, and actually I asked Anna and Raquel, okay, we have to have evidence that this, what we measure is true, and they really repeat the experiment giving N-acetylcysteine to the animals before they were injected, some weeks before. And the N-acetylcysteine is actually reducing the signal of ROS. So that was pretty convinced that this new molecule developed by Mike Murphy was a very good molecule in that sense. We really confirmed this by other methodology, more classical. But what was interesting is that is also cells having more ROS in vivo, liver, or heart have more mitochondrial DNA exactly reproducing what happened in the cells. And when NAC, as it did in the cells, reduced ROS, reduced also the level of mitochondrial DNA. More interesting, you take mitochondria from liver, you make them respire or make them to synthesize ATP in vitro, and you see that this response of having more mitochondria and mitochondrial DNA is needed to equalize the normal level of ATP production and respiration. If you prevent it, then your mitochondria now reveal a different capacity in ATP production, in function. So these secrets that read which mitochondria I have is slightly different, and how I have to proceed to really reestablish the wild-type level. So those are wild-type mitochondrial DNA anyway. So I cannot go because it's a very short time, but we found differences in the organization of the electron transport chain. This is a blue native. This is the complex one, entirely complex one, and one interacting with three. And the overall message is that having one DNA of the other readapt the electron transport chain system of macromolecules. This is a proteomic analysis of also complex 1, complex 4, or complex 3 of 12 weeks or 2 years of age, and you see that from 12 to 2 years it evolved. There is at the beginning there is less complex 1 as an example, per mitochondria as an example that they have different consequences in expression, and then react with age in a different way, behave completely different. So the idea is that the adaptation is tissue and cellular dependent, and the conclusion should be in the same, have the power that cells need. So there is also changes in glycolytic production of proteins, and in beta oxidation you have induced by the mitochondrial DNA, and there are changes also over the age. And what about function? This is a very interesting question, because it also tells us that when we analyze and study in vivo an animal from metabolic perspective, we shouldn't stick with one age, because it could really, you can extend the observation to every age of the animal. This is a logarithmic scale representing from 4 days after birth to 700 days of age. And we just have here is the relative function for either ATP synthesis or respiration of mitochondria isolated from liver. When the ratio is 0, because it's a logarithmic rate, is that they perform equally. And when the ratio is under 0, is that black 6 mitochondrial DNA perform better. And when it's over 0, is that NCB perform better. And you see that the situation is not constant. Perform better, black 6 after birth, and perform better NCB when they go older. Why? Because black 6 mitochondrial DNA lose function over the age, and this function is preserved by NCB. It's not that it's improving the function, it's that it's preserving the function. So not only preserve the function, but also preserve the same abnormal high level of ROS, while the other that started with low level really lose control and enter in a level of ROS that is, at least in liver, going probably in troubles. There are many aspects that change in terms of the structure of the mitochondria, being both healthy, I should insist that, and we analyze that by ultrastructure. There are also things that were driving me a little bit skeptic of all this analysis for some months, is that despite of these changes that we see, the animals, they weigh the same. They were not differences that we could really appreciate at the macroscopic level in the animal. So we did transcriptomic, and there was also distinguished in every tissue response of transcriptomic having one DNA of the other, so to the point that we could tell you only knowing the transcriptomic parameter which DNA has this sample. And the transcriptomic affect free radical scavenging, but also lipid metabolism and glucose metabolism. That was very interesting. And in particular, it was very relevant in liver that we show by transcriptomic an activation of lipid metabolism, both synthesis and degradation, a reduction in inflammatory response, and a reduction in glycolytic if you have NCB mitochondrial DNA with respect to black 6. There was also differences in the metabolite of the liver and the plasma and the serum, in which they separate perfectly mice having one or the other mitochondrial DNA. And the one interesting pathway that is affected is glutathione biosynthesis. So the overall idea is that at the level of the single cell transcriptomic, at the level of the organism serum, there are really, this animal is aware of the mitochondrial DNA that they have. There was changes in the proteomic, but I'm gonna go just to show the final results is still, is omic telling us the truth, or is omic just a gambling of numbers that tell us what we wanted to see? Because the animals having different metabolism at the level of omics, they don't show any problem at the level of the macroscopic animals. So Anna challenged the animals and gave it half a diet, and now everything was revealed. The animals that has a more active lipid metabolism get less weight than the animals that does NCB, than the animals that have less active lipid metabolism. And more interesting, when the animals stop growth, they do at the end of the 9-10 months, you don't need to give them high-fat diet to reveal immediately in chow diet a different way in the maintenance. So it's like we stop after the growth of the individual, we have a metabolic change, and this happened also in the mice. And then what was hidden, because the energy consumed to make body, was actually hidden the differences. This is because I insist that we have to follow the animal. Moreover, glucose metabolism was also affected. This is the GTT analysis. Moreover, if you follow the animal all over the age, you see that the median lifespan was extended having the foreign mitochondrial DNA with respect to the control. The maximum is not changes. And these two individuals have exactly the same age. So how healthy is the individual when they age? It depends on how handled the metabolism through mitochondria during the entire life. So this was confirmed by telomere attrition that is double in the animals that has black 6 mitochondrial DNA with respect to the NCV. It was confirmed with having less neoplasia in the liver, has a better preserved oocyte formation, better reducing the aging phenotype of the knee, and many aspects. And this is just the final slide, in which what we believe that happened is that the little bit more rust triggered mitochondrial biogenesis, triggered mitomesis, and triggered membrane recycling in a general way. This requires changing the metabolism, less glucose metabolism, and increasing the synthesis and degradation of lipids. And this is, in general, generating a protective phenotype in the animal facility. If you go to the wild, maybe NCB is not the one that you want because it's less efficient in terms of muscle power. And thank you very much. Again, sorry because I think that I took a little bit more time than I should.
Thank you, Toño. Very nice. So you are proposing that something that happens very early in the life of the animal impacts the whole lifespan of the animal. So that suggests there is some kind of imprinting mechanism early on that regulates what happens during the whole life. Do you have any idea about this?
Well, I'm proposing that, but not only that. One thing that we know for many other organisms, for example, for yeast, we know that yeast also age, and it can be counted the number of buddings. And if you stress not very much but sufficient with hydrogen peroxide when they are very young, they have a longer lifespan. If you do when they are already old, you don't have the extension of lifespan. So it is a general in live entities that the hormesis phenotype should be triggered when they're in the early stage of life. In addition, I think that one of the consequences of the hormesis mechanism is that they trigger the continuum non-specific reparent system. So you make membranes because you decided that you are really having a high chance to break membranes. You are against the problems that you have identified and many other insults that you don't have any way to test. So I think that it is set at the beginning, but has to be continuous all over the age. If you block it, at a certain point you don't have the benefits. I think that this is what we interpret the things here. But it's not only in mammals. I think that is more general.
Toño, I've noticed that the time frame in which these physiological changes occur in the mitochondria are in the order of months, right? The question now is, this is a long time in terms of the biology of the cell. To which extent these changes in the mitochondrial physiology require, or do not require, transcriptional changes in the nucleus?
So they for sure require.
They do.
And this is a fundamental hallmark. So what surprised me a lot is that we only put 2 tissues, but you go over many tissues in the cell. And you see that every cell is able to distinguish with a very tight and very clear response in the transcription program. So the cells know which mitochondria they have, that means which function of the oxyphos they have. You can really trigger the same thing, just not by genetically, also with pharmacological induction. You can trigger, but you have to be very careful because you can poison the cell, and you don't want that. But you can also trigger the response. I think that this is a fundamental reading out of the nucleus and making an idea which is my environment, which is my genetical complement in terms also of mitochondria, and how I should react. And if I can tell you, we have now challenged this response, putting the 2 DNAs in the same cell at the same time. The response is beautiful. Most of the tissues kick 1 DNA out. They can and they kick not randomly, they kick the one that but it doesn't fit very well with their function. And those cells that are unable to kick any of them, they get sick because they cannot adapt transcriptomic to both different demands of 2 different mitochondrial DNAs.
Hi, so in your experiment where you keep the same nuclei and you put different mitochondrial DNA, so a population geneticist or quantitative geneticist will say that this is a sample of n equals 2 from the unlimited possibilities of the type of combinations of nuclear DNA that one can make. So can you speculate to what extent this response can hold in a large number of possibilities of combinations of nuclear DNA with different mitochondrial DNA?
Okay, I think that is a very pertinent question. And allow me to say that we bring this to the extreme just to be able to show if there is something or we have to forget about this any longer. So the idea is that this situation that is inbred, so remember this nucleus is substantial homozygotes, it's not 100%, and this is not a natural situation. So this proves that this intertalk happened. But how this reflect in the population? So, and there are many questions that are coming here, and the difference between the 2 mitochondrial DNA that we have is equivalent to the difference between an African haplotype of mitochondrial DNA in humans versus an European. So we are in the range of the variability that is happening in the human population variability or in the general population variability. This has been analyzed beautifully in Drosophila. In Drosophila, they could do this kind of experiment that you are really requiring here, and it cannot be done in mice because in mice we don't have enough data of variability. And in Drosophila, they proved a substantial impact, not only of the variant of mitochondrial DNA, actually the interaction of the variants of mitochondrial DNA with the matched proteins in the nuclear DNA. And they can range from a completely sterility of males, having the females the maximum fertility in some combination, to really very dramatic and significant changes in the phenotype within the range of wild type. So the answer is, at least in flies, this is very relevant. The problem in flies is that they couldn't go molecularly to the level that we came here. So I think that the two analyses complement, and they extended for, at least for animals, that this is really going to be relevant and very likely in humans. In humans, think that how this substrate can really affect the expression or the diseases or the response to insults in the environment from different people. I think that this is the relevance for the study in human beings.
So if there are no more questions, we must move on. Thanks very much, Antonio. Thank you.
Thank you.
So we move to our third speaker, is Seamus Martin. He is full professor in the Trinity College of Dublin and a great expert in mechanisms of apoptosis, internationally recognized by his works related to death receptors and the signaling from death receptors and their relationship with different physiological and pathological processes. Thanks very much, Seamus, for being here and for accepting our invitation. Thanks very much.
Turns out it wasn't my computer. Okay, sorry about that. First time my slides have actually broken the data projector. Okay, I'll try to be quick. I'm not sure if I can fit it into 15 minutes, but I'll certainly try. I'll just kind of try to hit the key points. Before I start, I'd like to thank Isabel for the invitation and the other organizers. It's really nice to be here. And I also want to just mention the FEBS journal. I took over the FEBS journal about 3.5 years ago now. And if you haven't looked at our website, we publish in all areas of the molecular life sciences. The impact factor of the journal is almost the same as the JBC and rising. And essentially we're, I suppose, fulfill the remit of the JBC in Europe. And please, if you haven't submitted a paper to us, we'd certainly like to see your papers in any area of cell and developmental biology or biochemistry, signal transduction, immunology, and many other areas. And our advantage is that we don't charge any page charges. It's free to color every single figure in the journal if you want, and no publication fees of any kind. And we only publish about 10 to 12 papers per issue. So we all know this. In biology, how you view the problem very often dictates what answer you get. And I just want to illustrate this with Mount Rushmore. And this is another viewpoint, obviously, of Mount Rushmore that we don't normally think about. And in a few years' time, I guess it could look like this. So anyway, so I'm going to talk about death receptors. And these are molecules, if you hadn't heard of them, I'm sure many of you have. They're molecules that belong to the TNF family, and particularly two of them, FAS and TRAIL. They're cell surface expressed, and they recruit molecules, particularly this molecule here, procaspase 8, upon activation of the receptor. And there are literally more than 10 to 15,000 papers in the literature describing how engagement of these receptors promotes programmed cell death, apoptosis. So typically within a few hours of engagement of FAS or TRAIL receptors, cells very often, or at least that's what the literature would tell you, that cells die. And we know very well exactly how this happens. Caspase 8 is recruited to the death receptors via this adaptive protein FAD, and then caspase 8 undergoes catalytic activation to unleash its full protease activity, and then it can promote programmed cell death by activating other caspases either directly or through permeabilizing mitochondria, which also results in this caspase activation cascade. And as I mentioned, there's more than 10,000 papers in the literature really investigating how death receptors engage caspases and under what circumstances, and investigating whether molecules such as Fasertrail can be used to treat tumors. Now, the overwhelming perspective on these molecules is that they're death receptors. I've used the term several times, but there's another side to these molecules that I'm going to talk about today that really is very understudied. There's probably about a dozen papers in the literature on this aspect of death receptor engagement, and this is what really I'm going to focus on today. So over the years, so just to show you, I mean, it's certainly the case that in many cells, when you treat them with ligands for the Fasertrail receptors, within a few hours, you get these very beautiful apoptotic cells, and all of the features of apoptosis are associated. But there have been a series of papers over the last 10 years or thereabouts with these counterintuitive outcomes of Fasertrail receptor activation. So first of all, this about 10 years ago, Marcus Peters' lab showed that activation or treatment of cells with CD95 ligand, so FAS is also called CD95 receptor, so CD95 ligand induces motility and invasiveness of apoptosis-resistant tumor cells. There's a paper here from Anna Martin Villalba showing that CD95 can signal invasion of glioblastoma, and another paper from Marcus Peter showing that CD95 receptor is required on tumors and also for regrowth of livers after hepatectomy. So that suggests that FAS is somehow required for tumor proliferation and invasion, which doesn't fit with its role as a molecule that induces apoptosis in tumors. And there's also a series of studies showing that FAS receptor expression is highly upregulated in renal carcinoma, on glioblastoma, and a number of other tumor types. So again, that's not something you'd expect for a molecule that transduces death signals. You wouldn't expect tumors to upregulate these molecules. And there are also other papers, which I'm going to show you in a moment, on TRAIL. TRAIL also is required for invasion of a number of tumor types, including pancreatic ductal adenocarcinoma. And if you delete TRAIL receptor, these tumors fail to progress as aggressively as they normally do. And some data from my own lab a few years ago, we looked to see what would happen when we overexpress FAS on this transplantable tumor cell type, Lewis lung carcinoma. When you implant these tumors into syngeneic mice, they grow a decent-sized tumor in about a month. And contrary to expectation, when we overexpress FAS on these cells, the tumor growth was actually accelerated. Now, again, current views of FAS would predict that if you overexpress this molecule, that the tumors would be more readily cleared because they've been more susceptible to apoptosis, but in fact we get the opposite result. The tumors grow much more aggressively. These are tumors at the same age. These are the wild-type parental tumors, and these are the FAS overexpressors. And when we took these tumors out and measured cytokines and chemokines to try to kind of figure out what is it about FAS overexpression that made the tumors more aggressive, and we found that there was elevated levels of a number of different cytokines and chemokines. And of course, most of you know this, but I just want to put up this slide just to remind you that cytokine and chemokine production harnesses essentially all aspects of the immune system. So these molecules not only recruit immune cells, for example, chemokines are very important recruiters of macrophages and phagocytes and T cells, but they also modulate the activity of immune cells. And obviously, tumor inflammation is a hot topic for the last number of years, and I'll get into some of the reasons why that is in a few moments. And these tumors are extremely heavily infiltrated by macrophages and neutrophils, and that's typically a bad sign. In about 90% of cases, when there's a lot of immune cell recruitment, particularly innate immune cells, that's a bad prognostic indicator for solid tumors. Rather than clearing those tumors, the immune system in these situations seemed to be helping the tumors. So it seemed that inflammatory signature that possibly is turned on by FAS on these tumors is helping these tumors to become more aggressive. But as I mentioned, that role, that potential functional outcome of FAS receptor stimulation really has been not explored, And so that's something we wanted to look at. And there's some data on trail inducing metastasis and progression of pancreatic ductal adenocarcinoma. So the question was, how might faster trail benefit tumors? I've already said it there now. We thought that faster trail expression in tumors might be encouraging or fostering an inflammatory environment in tumors, and that's what we started to investigate about 5 years ago now. So I mentioned earlier that FASN trail belong to the TNF receptor subfamily, and these molecules in the cell death field, they're typically viewed, as I've said repeatedly a few times, as molecules that promote apoptosis. But they also can be, they can also drive NF-kappa B activation. And again, in the cell death field, NF-kappa B activation is very often thought as an anti-apoptotic signal, but of course that has a much more important role than that. In the immune system, NF-kappa B is essentially a master regulator of multiple pro-inflammatory cytokines and chemokines. And again, there's this kind of different worldview depending on whether you're coming at the death receptor family as an immunologist or as somebody who's interested in cell death control. Of course, immunologists know TNF very well as a very, very aggressive pro-inflammatory molecule. So in fact, its potential role as an apoptosis-inducing molecule is quite a minor one. In fact, most cells do not respond to TNF by dying. They respond to TNF by essentially instigating a very potent pro-inflammatory response. So this is what happens when you treat cells with TNF within a very short period of time. This is just looking transcriptionally what cells are making. These are cells that don't die at all in response to TNF treatment, and that you get very, very strong upregulation of many, many chemokines and cytokines within just a few hours of TNF receptor stimulation. And inflammatory molecules, cytokine and chemokine production in the tumor environment, we now know is very detrimental. It's not a good sign, and it's something that generally is to be discouraged. And this is for a number of reasons. So tumors themselves can initiate inflammatory cytokine production. In fact, they almost certainly must do this in order to build up this macrophage and neutrophil environment, this inflammatory environment that starts a process called smoldering inflammation. So somehow tumors manage to secrete cytokines and chemokines of various types, the green arrows here, and that draws in macrophages and neutrophils. And then the macrophages and neutrophils, particularly macrophages, then may contribute to the tumor by producing angiogenic cytokines or cytokines that may actually suppress cell death within the tumor, or cytokines that further boost tumor inflammation. And also these macrophages may display checkpoint inhibitor proteins that suppress CTL responses, so they protect the tumor from CTL killing. They may also deplete critical amino acids that suppress T cell function, secrete anti-inflammatory cytokines such as IL-10 and TGF-beta, and also molecules as IDO, which also can induce Tregs and suppress T cells. So on multiple levels, we now know that if tumors manage to attract the attention of the immune system and particularly induce a wound healing type response in the tumor, that these macrophages that are recruited to the tumor can actually be basically tumor protective by producing wound healing cytokines, proliferation inducing cytokines, as I've mentioned, produce angiogenic factors, and multiple other molecules that actually suppress immune responses within the tumor. So inflammation in the tumor environment is not a good thing, but despite that, we really don't know how inflammation is triggered in the tumor environment. Now, there's a number of different candidate ways that inflammation may be instigated, and I've listed some of them here. But there is no doubt that many solid tumors, if not all solid tumors, manage to somehow trigger an inflammatory response. And there's two major ways we know that inflammation is induced, either by pathogen-derived molecules, which are called PAMPs, or pathogen-associated molecular patterns. So effectively, if you have an infection, then you would expect to see inflammation. Or if you get an extreme amount of cell death, so necrosis, you can get release of molecules that have been called DAMPs, danger-associated molecular patterns. But these are typically associated with necrosis. So how tumors manage to trigger an inflammatory environment is still an open question. And there's a number of candidate ways. So when you have a pre-existing inflammatory condition, so for example, individuals with Crohn's disease have a greater susceptibility to getting tumors in the colon. If there is an infection, then of course that can drive an inflammatory environment. So if you have persistent infection associated with viral infection, hepatitis B infection, for example, can predispose towards liver cancer. Certain oncogenic mutations may result in constitutive activation or constitutive production of cytokines, and so on. So really, our question was whether upregulation of death receptors by tumours may be a means of instigating tumour inflammation in certain circumstances. So a number of years ago, we embarked upon this, and we published a paper actually on this, so I'm going to go through the data very quickly. So it's one of those experiments where when you have the idea and you go back and test it in the lab, and this is quite rare, the very first experiment works and they keep on working. And sometimes things just work, but of course a lot of the time they don't work. So we know very well, as I've said now a number of times, that when you treat cells with FAS or TRAIL, typically you get a lot of cell death. But of course there's always surviving cells. And when we did the very first experiment, we measured cytokines in the medium, and we found very easily that FAS and TRAIL receptor activation not only killed cells, but also induced a lot of cytokines and chemokines. All of the cytokines and chemokines, in fact, that TNF receptor activation produces. And we saw this in transformed cells as well as primary cells. And also we injected FAS in vivo into mice. Again, very easily we saw within a few hours very high levels of circulating chemokines and cytokines in the bloodstream of these animals. So there's no doubt that FAS receptor ligation, in addition to being a pro-apoptotic stimulus, can also result in cytokine and chemokine production. So FAS acts as a pro-inflammatory factor as well as a pro-apoptotic factor. And there are some cells, in fact, and this was the surprise, there were some cells that would have been considered non-responsive to FAS that we tried. So when you treat them with FAS, they don't die at all, and they would be typically regarded as non-responsive. But of course, they are responding, they're making cytokines and chemokines. So we found many different cell types as well that didn't die at all in response to FAS or TRAIL, but made lots of cytokines and chemokines. So somehow they'd managed to rewire their receptor to selectively activate the inflammatory output as opposed to the cell death output. Now, I'm going to have to skip through this quite quickly, but you already get the gist of it. I'm really focusing on the inflammatory output. So more recently, we showed that TRAIL receptor expression also results in a very similar inflammatory phenotype. Again, you can get death, but also cytokine production in response to TRAIL receptor ligation. And again, we found a number of cell types where TRAIL receptor engagement barely induced cell death, but induced robust secretion of cytokines and chemokines. This is a nice example here, H29 cells. Untreated cells have absolutely no interleukin-8, but within a few hours of treatment with TRAIL, the cells have no apoptotic phenotype whatsoever, but if you stain for intracellular IL-8, and of course you look at the medium, you find lots of IL-8 being produced. So death receptors don't just induce death, they also induce inflammation, and that can occur in tandem, or it can be actually, you can get the inflammatory output without the cell death output. I'm going to skip that, I'm going to skip that. So there's multiple consequences of inflammatory cytokine production in the tumour environment. I don't have time to show you this, but I just want to show you that the amounts of cytokine and chemokine that are being produced upon ligation of these receptors is actually physiologically relevant. So we did some chemotaxis assays. So you take basically supernatants from untreated cells or TRAIL-treated cells, and then use either primary human neutrophils in a chemotaxis assay. And you can see that the neutrophils are labeled here in green, and using a Boyden chamber assay where the neutrophils are put in the upper chamber and the supernatant in the bottom chamber. Very few neutrophils migrate towards controlled supernatant, but when you've taken supernatant from TRAIL-treated cells, you get a lot of neutrophil chemotaxis and also monocyte-like cell chemotaxis. And the neutrophil chemotaxis is, sorry, that's also the same with FAS. The neutrophil chemotaxis is largely interleukin-8 dependent, as we would have expected. So interleukin-8 levels that are produced in response to TRAIL or FAS ligation are sufficient to recruit primary human neutrophils, basically the source of those chemokines. And MCP1, which is a monocyte chemotactic factor, is more important for monocyte macrophage recruitment. So basically what I've told you so far is that Faster trail receptor ligation can result in apoptosis, and even in those circumstances, we find that there are cytokines and chemokines being produced. But we can also find cells where they don't die whatsoever, and they preferentially make cytokines and chemokines. And of those, interleukin-8 levels are instrumental in recruiting neutrophils, and MSP1 is instrumental in recruiting monocytes or macrophage-like cells. So more recently, we've been looking at how the faster trail receptor promotes an inflammatory signature. And again, I'll go through this very quickly. But TNF receptor signaling, in terms of how TNF induces NF-kappa activation, has been studied very intensively for actually almost 20 years now. And molecules that are critical here is RIP kinase 1. RIP kinase 1 is recruited directly to the TNF receptor, and it becomes linearly ubiquitinated and ubiquitinated on other sites. And that results in activation of either the NEMO IKK complex or the TAC-TAB complex, and they either individually or collectively activate NF-kappa B by triggering degradation of I-kappa B. So RIPK1 recruitment is really important in TNF receptor-induced NF-kappa B activation. But how RIPK1 is recruited to the FAST receptor really wasn't clear. And RIP kinase recruitment to the FAST receptor has been recorded, in very few papers over the years, but it's not really clear whether it's recruited directly to the receptor or via this adaptive protein FAD, or possibly via caspase 8. It certainly wouldn't have been anticipated that it bound to caspase 8, and so the next experiment we got was very surprising. I skipped through data early on to show you that predictably, so when you treat cells with trail, they die in some cases. And then if you add a caspase inhibitor, this molecule here is EVAD-FMK, you completely suppress death by blocking caspase 8 activation. And we had found that predictable, we really expected this result. We did not block cytokine production if we blocked caspase 8 activity. In fact, if anything, you can see here in many cases we got more cytokine production or chemokine production when we treated cells with trail in the presence of caspase inhibitors. So that seemed to suggest to us that you didn't need caspases for trail or fast-induced cytokines or chemokines, because if you blocked caspase activity, you blocked death, but you enhanced cytokine production. So we got a big surprise then when we actually knocked down caspase 8. We got the opposite result. This is what we published a few years ago for FAST. So if you knock down either FAD, the adapter for caspase 8, or caspase 8 itself, and you measure cytokine and chemokine production, you completely abolish cytokine and chemokine production in response to FAST, and also data I'm not showing you here to trial. That was a really surprising result. So if you inhibit caspases with a caspase inhibitor, you enhance cytokine or chemokine production, but if you knock down the caspase, you completely block cytokine production. We don't get the same with TNF. So in green here is caspase 8. When we knock down caspase 8, we don't interfere at all with cytokine and chemokine production. But again, you can see that if we knock down caspase 8 with trail, we completely suppress cytokine production. So that's suggested that how FAST or trail induced cytokine or chemokine production is very different to the way the TNF receptor worked. And to cut a long story short, I'll just show you again that our knockdown results were diametrically opposed to our caspase inhibitor results. So again, if we inhibit caspases, we get more cytokines in response to trail. If we knock down the caspase here with two different caspase 8 oligos, either alone or in combination, we completely suppress cytokines. So long story short is that caspase 8 is essential for TNF or fast-induced cytokine production. If we CRISPR out caspase 8, so we make caspase 8 knockout cells, we suppress trail-induced cell death completely, and also suppress trail-induced cytokine and chemokine production. But then we restored a catalytically dead caspase 8 molecule into these cells. So we knocked the caspase 8 and restored with a catalytically inactive caspase 8. This is in blue. So this caspase 8 molecule cannot restore cell death in response to trail. Whereas when we put in wild-type caspase 8, you can see here it restores caspase susceptibility to trail in the caspase 8 knockout cells. But here in blue again, the catalytically inactive caspase 8, so the enzymatically dead caspase 8 can restore trail-induced cytokine production in caspase-8 knockout cells, which was a very surprising result. We have other controls to show you that it works also with FAS. So how RIPK1 is recruited to the TNF receptor seems to be very different to how it's recruited to the FAS receptor. In the case of TNF receptor, RIP can get directly recruited to the receptor, whereas, and I don't have time to show you this data, but FAD caspase-8 are required for recruitment of RIPK1. So in this case, caspase-8 is not acting as a protease, it's acting purely as a scaffold protein. And if you want to know more details, the paper was published early this year, but this is a little bit of the proof. When we do immunoprecipitation of either the trail receptor directly or the caspase-8 molecule to look at the details of complex assembly, when we inhibit caspase activity, we get far better trail or RIP kinase 1 recruitment to the receptor. And in caspase-8 null cells, so these are caspase-8 null cells, when we pull down the receptor, essentially nothing else comes to the receptor except for FAD, the adaptive protein. FAD comes and nothing else. So I know this is very fast to show you all that data, but essentially this is the bottom line. This is what happens when you engage the trailer FAS receptors. Basically, you get a complex that's formed of FAD and caspase-8, and then an additional FAD molecule binds and recruits RIP kinase 1. And then this complex can disengage to form what we've called a fadisome, which promotes NF-kappa B activation and not cell death. So this is how the TNF or FAS receptors promote inflammation. And I don't really have time to show you the last bit of the story, but the point here of this last bit of the story was that in fact there are many caspase-8 mutations have been found in tumors over the last 10 years, and all of these caspase-8 mutations have been considered to inactivate the protease activity of caspase-8 in order to block apoptosis. And we've been looking at these mutations, and in fact this is wild-type caspase-8 activity, and many tumors have elevated caspase-8 dependent NF-kappa B activation or inflammatory activity. So they carry caspase-8 mutations that are considered loss of function, but in fact in terms of their ability to promote cytokines, many tumors contain caspase-8 mutants that actually are better with this inflammatory function of caspase-8. So this scaffold function of caspase-8 is actually enhanced by mutation of caspase-8 in many tumor types. And this is essentially the bottom line. We think that many tumors upregulated FAS or trail not to increase their susceptibility to cell death, but in fact to facilitate the production of inflammatory cytokines and chemokines. And one of the ways in which they turn off the cell death axis is by acquiring mutations in caspase-8 that inactivate the protease function of caspase-8, but turn on its scaffold function that's important for NF-kappa B activation. And that's the three messages I've said today. Trail and FAS can also promote inflammation, and that might explain why they're amplified or overexpressed in certain tumor types. Caspase-8 plays a key role as a scaffold rather than a protease in promoting FAS or trail-induced inflammation. And many tumor-associated mutations in caspase-8 inactivate the pro-apoptotic function of caspase-8, but turn on the pro-inflammatory function. And I want to thank Sean Cullen, a great PhD student in my lab, who's now at Genentech, and Conor Henry, another PhD student in my lab, who did all of the work I've showed you today. And I thank you for your attention.
Wonderful talk, congratulations. So maybe two questions. Do you see the cells that die versus the ones that don't die, do they have any difference in STAT3 activation levels, for example?
We haven't looked at STAT3, but we think that, for example, FLIP levels are very important in this case. So there's a molecule called FLIP that I haven't talked about today, but it's a caspase-8-like molecule, but it has no catalytic activity. And cells with high FLIP levels tend not to die. and those with low FLIP levels tend to die extremely readily. So we think FLIP is important, and also molecules called IAPs that also can recruit to the receptors. So this may dictate whether or not cells live or die in response to faster trail receptor, but we haven't looked at STAT3.
And another question, if you looked in your in vivo studies, how T-cell infiltration correlated with macrophages?
We haven't looked at that yet. What we are doing currently is we're making caspase-8 null tumors and death receptor null tumors to see if this influences macrophage recruitment to the tumors. But we haven't looked specifically at T-cell recruitment versus macrophage recruitment. Sorry.
Hi, very impressive, very nice. I probably miss, you probably said, the models in vivo that you used, you injected cells, tumor cells, and in nude or in regular mice?
These are in wild-type mice.
Yeah, so did you have a check in the early stages if the inflammatory response in the very early stages after is different, so you can calculate if you have more inflammation or less after the injection?
So this is something we're doing now. So we don't like this overexpression model. So we're actually deleting, in these tumor cells, we're deleting the death receptors, individual death receptors, as well as caspase-8. And they're looking at this more finely, this recruitment. So we haven't done any kind of fine kinetics yet.
Okay.
I got a very basic question. I don't quite understand why creating a pro-inflammatory environment and attracting macrophages and so on would benefit the tumor in some way. How does the tumor benefit itself from creating a pro-inflammatory environment?
Okay. No, it's a very good question because I think immunologists for a long time were very puzzled by the fact that most tumors have recruited a lot of immune cells, but the immune system isn't attacking the tumor. So it is very well established now that the type of immune environment that exists around tumors is more a wound-healing environment. Because of course the immune system has two major functions. One function is to obviously repel infection, but in most cases when you get infection, you've got damage, and that's why you get infection. You breach the skin barrier or one of your other major barriers, and that damage needs to be repaired. So in fact, the immune system is very good at coordinating the repair of tissue. And to get repair of tissue, you need to remodel a blood supply, you need to provide proliferative cytokines, and also basically provide the goodies required to repair tissue. So the immune system is very, very good at inducing repair, and inflammation, that's one of its major roles, is to coordinate tissue repair. So it's that tissue repair function of the immune system that tumors are preferentially inducing. So they're not inducing the aggressive, let's say, antimicrobial type function. So it's very, in fact, you would expect so tumors mimic wounds essentially. That's what they do. And so the immune system doesn't know that this is a wound. And macrophages, when they're recruited, are coordinating this wound repair process and also suppressing the function of the immune system that could kill the tumor cell. And that's why if you turn off these functions, so it's now known that by inhibiting these checkpoint molecules that are present on wound-healing macrophages, you can then reactivate the more aggressive cytotoxic functions of the immune system.
Seamus, I understand that if NF-kappa B is the main signal responsible for the high cytokine or chemokine production, is the regulation is only at the transcriptional level?
As far as we know of, predominantly transcriptionally, yeah, we see these are newly synthesized chemokines and cytokines. So I think it is predominantly at the transcriptional level.
Mainly transcriptional.
Yeah.
Last and short and very naive too. I gather that you think this mechanism does not operate in the liquid tumors.
It may operate in liquid tumors in environments where liquid tumors are at sufficiently high density that they can benefit from, let's say, cytokine, chemokine production. So in the bone marrow, for example. So it could actually operate in liquid tumors as well. We know that certain leukemias and lymphomas, when you stimulate with faster trial, they also make inflammatory cytokines. So it's possible that in liquid tumors, they may well benefit.
So we thank to the three speakers for showing us so novel and exciting data, and thanks to all of you for your attendance.